Hi, my name's Will Krawczyk, Scientific Services Manager at Chemical Computing Group. Today I'd like to show you how to find key contacts between a protein-protein interface for a specific loop or region or a given residue. So let's get started. In today's example, we're going to be analysing the contacts of an antibody-antigen complex, in this case interleukin-13 on the right-hand side, bound to an antibody on the left, although this same workflow can be applied to any protein-protein or protein-ligand complex. In this case, our system has already been prepared for interactive analysis using the QuickPrep application, and you'll notice that our antibody has been annotated according to the IMGT numbering scheme. This displays the light chain in a slightly lighter colour, heavy chain in a darker colour, and it also renders our CDRs. So for the light chain, these are rendered in purple, allowing us to identify L1, L2 and L3. For the heavy chain, H1 and H2 are rendered orange, with the third CDR of the heavy chain coloured red to remind us that this is more variable than the others. As part of this annotation, sets have been generated and populate our system manager. So these are collections of atoms such that they can easily be selected for use later. With our system prepared as we'd like, we can now analyse these interactions, and we can do that using the Protein Contacts panel, which comprises three main sections. The top allows us to define the sets of atoms we'd like to analyse. The middle provides the detail on these contacts, and the bottom allows us to filter. By default, both set A and set B are set to receptor, but if I just click the pull down, you'll see that we can analyse selected atoms, residues or chains, or I can select from the PDB tag itself, so for example the light chain or the heavy chain. Today though, I'm going to leverage the sets or the collections of atoms that we generated when we annotated our antibody and that populate the system manager. Here I'll select the antibody for set A and I'm going to select the antigen for set B. I'll click search to return the interacting pairs. So here, reading from left to right, for the first line I see that we can identify the type of interaction. In Mo, 6 types of contacts can be identified, including H-bonds, metal, ionic, arene, covalent, and distance type interactions. So here I see that we've got a salt bridge implicating glutamine in position 1 of set A, and asparagine in position 113 of set B. I get a readout in terms of the energy and the distance, and I can see where the interaction occurs. Here the first character represents the contact in set A, and the second for set B, and if I just raise bubble help, is telling me that lowercase b indicates that it's a backbone-backbone interaction. I also get a readout for the frequency. This means the total number of interactions that have been aggregated into a single line. To analyse this interaction, it might be useful to sort through our data, and I can sort by any of these column headers. Perhaps a good place to start would be to analyse with respect to energy, such that the strongest interactions are now displayed at the top. Here I find that we've got several salt bridges that implicate negatively charged residues on the antibody, and arginine and lysine residues on the antigen. Below that I see that we've got some pretty good H-bonds between these residues, but by default you might notice that distance type interactions are toggled off. That's deliberate because typically there's a lot of them. If I just toggle those on and scroll down my list, I see several of these distance type interactions that are typically weaker, but as I say, there's lots of them, and that means that together they can become quite important. I can also select any of these interactions from the panel so I can view them in the main Mo window. If I just go ahead and select our strongest contact, isolate and center, I'll just move that panel to one side, and we see that our pair of contacts displayed with a halo surrounding them. You'll also notice that the classical hydrogen bonds have been calculated and annotated, shown by these dotted lines and the cylinder in the middle. The size of the cylinder is important, whereby the longer the cylinder, the stronger the bond. Next, what I'd like to do is focus in on a specific region so that we can see the key interactions for that region. I'll just go back to our protein contacts panel and I'm going to update our set A so that we can analyze interactions with CDR loop 1. I'll click search to update that list. Today we've been analysing our contact list, but you might notice that I can toggle this to view this as a plot, such that the x-axis represents the residue sequence for set A and the y-axis for set B. Here I'm looking for several dots on the same horizontal or vertical line, meaning that that particular residue might be implicated in several interactions and therefore may be important. I see such a scenario just here, so if I just drag a selection box over those and toggle back to contact list, I see that the residues that I've selected all implicate phenylalanine in position 31. In this case, what I'd like to do is further focus in on these interactions. So I'll use the filter here and type in the residue name and number and hit return such that I can just isolate those specific interactions. So here I can see that phenylalanine in position 31 is actually making 14 interactions with lysine in position 97 alone. By default, these contacts have been aggregated. So if I want, I can switch this to type so that I can differentiate between the distance type interactions and the arene type interactions. And if I opt to have no aggregation on at all, each of these separate interactions is now displayed on a separate line, also indicating the atoms implicated in that. So if I just toggle back to the residue view in terms of aggregation, I can see that where I see a frequency of 14, it means that this pair of residues is making 14 distinct interactions between pairs of atoms. So let's view these in the main Mo window by hitting Site View so that we isolate the selected contacts together with anything within 4.5 angstroms. I get a readout on the footer here in terms of the total area of those selected. And should I wish, I can also visualize the surface between our sets. And I'll do that by clicking Show Surface. Here I see in pink the surface corresponding to the selected contacts. So should I wish, what I can do to isolate the contacts for lysine in position 97, I'll just have that as the only one that's selected, and they're displayed in pink in the main Mo window, allowing me to analyze these in greater detail. So in this case, we see phenylalanine is creating several distance interactions with the antigen. Although none of these interactions are particularly strong, the high frequency of these interactions suggests that this could be an important residue in the binding of our antigen. And with that, it brings us to the end of today's session, where we use the protein contacts application to help understand key contacts between first a protein-protein interface, before analyzing interactions with a specific region, and finally interactions with a specific residue. If you have any questions, please do reach out to us at support@chemcomp.com.